So let's see how to answer some GCSE Biology exam questions on investigating osmosis in plant cells. This set of questions is practical based. It requires us to know how to perform the investigation and analyse the results. It then asks us to link all of this to the biological knowledge of osmosis. The question starts by introducing a practical to investigate the effect of different concentrations of sugar solution on pieces of carrot. The method is described in bullet point form, starting with weighing 5 pieces of carrot and placing them into different test tubes. Various solutions are added to the tubes, ranging from just water to 0.8 moles per decimeter cubed sugar solutions. The tubes are then left for 2 hours before the carrots are removed, dried and reweighed. The percentage change in mass of each carrot piece is calculated, and the diagram shows this information visually. The results are shown in the table here as the percentage change in mass for the carrots in each of the 5 solutions. Our first question asks why students calculated the percentage change in mass of each piece of carrot. We can see from the diagram that the carrot pieces are not all the same size, so they're likely to be different masses to start with. Therefore, by calculating the percentage change, it allows us to compare the change in mass between the different pieces of carrot. Otherwise, if we just recorded the change in mass, we'd struggle to compare them unless they'd all started with exactly the same mass. We now have to plot the data as a graph, choosing an appropriate scale for the y-axis and drawing a line of best fit. We can see that the percentage mass change data range from -15 up to 24. So if we assign each major block on the graph paper as 5 percentage points, then we can fit all of the data onto the graph paper provided, while producing a graph that is large and clear enough to use. As a guide here, aim to use at least half the graph paper provided. When plotting data onto the graph, we must make sure that we use clear crosses to mark each point. We use crosses so that the examiner can see clearly where the points have been placed, and therefore give us marks for them. If we only mark our points with dots, then they may not be clear enough for the examiner to see, so you'll lose marks even if they're plotted correctly. Correct plotting means placing your cross within 1/2 of a small square of where it should be. This doesn't give us much room for error, so you'll need to be very, very careful. Now we have to draw a line of best fit. Be careful here too, because lines of best fit do not have to be straight. It can be a curve of best fit if that's what the data suggests it is. Just make sure the line is single and smooth. Next, we have to use the graph to estimate the concentration of sugar solution inside the carrot cells. This will be the sugar concentration for which there is no change in mass. We have to use our graph to see where the best fit line crosses the concentration axis. If I zoom in, we can see that the sugar concentration which produces no change in mass is here at 0.42 moles per decimeter cubed, so that's our answer. Next, we're asked to explain why the mass of the carrot in the 0.6 moles per decimeter cubed solution has changed. Let's quickly revise osmosis for a second. Osmosis is just a scientific name for the diffusion of water across a selectively permeable membrane. Remember that water molecules are very small, containing just an oxygen and 2 hydrogen atoms. This means they can easily pass through many membranes that other large substances can't. The cell membrane is one such membrane. Water will move across a cell membrane down a concentration gradient, which means if the concentration of water is higher on one side of the membrane than it is on the other, water will move from the region of high water concentration to the region which is lower. This cell has been placed into either pure water or a very dilute or hypotonic solution. The water concentration is higher outside the cell than inside, so water moves into the cell. In fact, so much water moves into the cell that the cell swells. The cell becomes turgid. This cell has been placed into a solution which is equal in concentration to that of the cell's vacuole. The cell and the solution are isotonic. Here, water will move into the cell, but it will also move out of the cell at the same rate. We say that there is no net movement of water and the cell remains flaccid. The last cell is in a very concentrated or hypertonic solution. The concentration of water is very much higher on the inside this time compared to the outside. A large volume of water will leave this cell, causing the cell to shrink. In fact, the cell shrinks so much that the cell membrane becomes pulled away from the cell wall, remaining attached in only one or two places. This cell has become plasmolyzed. So going back to the question, if we're going to explain why the mass of the carrot changed, we need to start by saying how the mass of the carrot changed. So we need to write the mass of the carrot decreased. Now we need to explain why this happened. It happened because water is lost from the carrot due to osmosis. And now we need to explain why this happened. This happened because water moved out through the partially permeable membrane. But why? Because the concentration of the water inside the cells is greater than the concentration of water outside the cells. So can you see how I managed to extend my answer from being a simple one-sentence answer to being a full answer worthy of the 4 marks? Do you all remember playing the why game as a child? Maybe you've got younger brothers or sisters that still play it with you. It's when you ask someone a question and as soon as they answer it you say why in that really whiny voice. And then they answer that and you say why again. And this carries on until usually the older person becomes annoyed. Well, that's what you need to do in exams. Play the why game with yourself. Imagine that really irritating 5-year-old sitting beside you screeching why. It works really well. Listen. Why is the grass green? Because the cells in the leaves contain a green chemical called chlorophyll. But why? Chlorophyll helps the plant to capture light energy from the sun. But why? The plants need this energy to turn carbon dioxide in the air into sugars. But why? They use the sugars as food to allow them to grow. You see, you can easily turn a short answer into a longer, much more detailed answer. Another trick is to use the word because at least twice in your answer. This will force you to expand, allowing you to really show off what you know. In the final question, we're told the student repeated the investigation with boiled carrot, but this time none of the masses changed, and we're asked to suggest why. A suggest question. These are usually the hardest type. It's asking you to use your knowledge to come up with a reason why. Let's think about osmosis again. We understand that the small water molecules can freely pass through the selectively permeable membrane, but the large sugar molecules cannot. So far, so good. But what happens when we boil the carrot? The membrane is destroyed by the high temperatures, just like enzymes are. Now, not only can the water pass freely across the membrane, but so can the sugar molecules. So now the concentrations of both sugar and water equalize. There's no region of high or low water concentration, so there's no net movement of water. So osmosis has stopped. This question is only worth 1 mark, so we don't need to go to town on the answer. We can just write that the carrot membranes were destroyed. So that's it for now. Keep watching my other videos for some great science content, or follow me for updates or revision tips at plutoniumscience.com. Or you can follow me on Twitter at PU94Science, or I'm now on Instagram. Keep up the revision and good luck with the exams. Take care.